Hi, everyone, and welcome to the Winehouse seminar. Today's seminar is going to be on Introduction to FMRI, and the speaker for today is Kaisu Lankinen. We'll be covering a very basic introduction to FMRI. So if you're someone who has been thinking about doing a project in FMRI and wanted some sort of brief introduction to it, I think you've come to the right place. Yeah, and the Winehouse seminar series is like a sort of a weekly or a bi-weekly series. If you're not subscribed to the mailing list, I highly encourage you to do so. We send updates there with regards to the seminars. The next one is going to be on batch processing. Yeah. So for now, let's get into the seminar on Introduction to FMRI by Kaisu. The floor is yours, Kaisu.
Thank you, Mainak. So hello, everybody. I'm a postdoc in Professor Jyrki Ahveninen's Auditory Cognition Lab, and I'm going to tell about a basic introduction to functional magnetic resonance imaging. Before I start, I want to acknowledge these people for sharing their slides and pictures, especially Yingyuan Cheng and Saskia Bollman. So fMRI gives us spatiotemporal information of brain activity. For example, here on the right side, you can see those yellow-orange blobs on top of anatomical MRI. And those tell that something is happening probably in the auditory areas. So with fMRI, we can make inferences what brain areas are engaged in some sensory or cognitive tasks, or we can, for example, measure functional connectivity between different brain areas. Here is a comparison for different functional neuroimaging modalities. We can see fMRI here in brown color, and we can see that the spatial resolution of fMRI it's a bit better than, for example, for MEG or EEG. With fMRI, we can get a millimeter spatial resolution, and nowadays we can even get below 1 millimeter in fMRI too. On the temporal side, fMRI is a bit slower than, for example, MEG, EEG, or neurophysiological methods. In fMRI, we are talking about on the order of seconds. But that's usually adequate for most of the tasks. So today I'm going to talk first about the neural basis of fMRI signal and bold contrast. Then we will look at the acquisition of MRI images, and then we will go through some signal and noise characteristics of fMRI, and finally we will look at some typical fMRI experiments and how to analyze them. So fMRI signal is based on changes in metabolic activity of brain when different parts of the brain are active. fMRI signal is not a direct neural signal, but it reflects neuronal activity via hemodynamics. And that's why we call fMRI signal as bold, blood oxygen level dependent signal. So the basic idea is that when certain part of the brain is active, there is a need for more energy into that area. So there's a need for cerebral blood supply into that area. And then oxygen consumption increases, also cerebral blood flow increases, and cerebral blood volume increases. Here's a picture of vascularization of the brain. Red are arteries and black are veins, and those very small veins, capillaries, between those they bring glucose and oxygen to neurons. So basically, in bold contrast, we take advantage of the fact that the hemoglobin has different properties when it's oxygenated or carrying oxygen, or when it's deoxygenated. Oxygenated hemoglobin is diamagnetic. It means that it has relatively little effect on the magnetic field, and deoxygenated hemoglobin is paramagnetic. It means that it introduces a local inhomogeneity in the magnetic field nearby it. So when we are at rest, there is more deoxygenated hemoglobin, relatively more in the blood, in the veins, and MR signal is smaller. But when there is some brain tissue becomes active, there is more oxygenated blood coming to meet the needs of the area, because when the neural tissue is active, it consumes the oxygen, and then it becomes deoxygenated. So basically what we can see in bold signal is that the ratio of these oxygenated and deoxygenated hemoglobin is changing, and that we can detect in the fMRI signal. This hemodynamic response to increased activity can be described with this kind of hemodynamic response function. It usually looks like this. So there is one prominent peak that happens around 7 seconds after the stimulus onset, and that is the peak is caused because there is more blood flow that brings more oxygenated hemoglobin to the area, and that the compensation is more than the consumption of oxygen. So that's why there is a peak. There are also two smaller peaks, initial dip and post-stimulus undershoot. The mechanism of those are not so clearly understood, and they might be variable between different subjects or tasks, but the initial dip is commonly thought to be because of the initial consumption of oxygen is increased before the new fresh blood kicks in, and also maybe the cerebral blood volume is increased, and the post-stimulus undershoot is probably caused by the prolonged activity or consumption of oxygen, or it might be also related to the ballooning of the vessels as a response to this peak. But in any case, the accurate shape of this hemodynamic response, it can be different for different subjects or even different brain areas, and it can change according to the task or different types of tasks. Next, we are going to look at the acquisition of fMRI signal. I will very briefly go through the basics of how MRI signal is formed. So in MRI, we typically use hydrogen nuclei, meaning single protons of water molecules, because there are plenty of them in the human brain, and those are the main source of signal in MRI. The nuclei, they possess a spin, and when the nuclei they are outside of magnetic field, the nuclei are, or those spins are into random directions. But when they are brought to the external magnetic field, like in the MRI scanner, those protons, they will align parallel to this external magnetic field that you can see in the figure B. Then slightly more spins are directed parallel than anti-parallel, as you can see in the figure, and that's why there is a net magnetization vector that points towards the parallel to the external magnetic field. What happens next in figure C is that we give a radio frequency pulse to the brain tissue, and that causes the net magnetization vector to flip with respect to the external magnetic field that you can see in the C, where the direction of these nuclei are now horizontal. And after the RF pulse, the spins, they start to return to their original direction, and simultaneously they emit a signal that can be detected in the scanner. So these two pictures, they show the process when the net magnetization vector returns back to their original position. There are typically two times constant that are used to describe this process. T1 describes the recovery of the longitudinal net magnetization, meaning the parallel to the external magnetic field, and T2 is related to the transverse net magnetization, so the other component. So this pure T2 would be because of random interactions between different spins, but there are actually other effects that affect it too. For example, the external magnetic field, it can be inhomogeneous. There can be some susceptibility differences in tissue boundaries. There can be metal in the body. So the some of these effects affecting the T2 are called T2 star. So this is combination of all these effects that affect the relaxation to the original position. Now, importantly, how this relates to fMRI, the bold signal, is that this hemodynamic response is seen in this T2 star as local susceptibility change. So we can detect that. So the deoxyhemoglobin, it shortens the T2 star, and then areas where there are more oxygenated blood, they will show up in the T2 star weighted images. here is some basic terminology of MR image acquisition. So field of view, it describes how big portion of the brain we can acquire, and the matrix size, it describes the dimensions of the slices, and these two together with the slice thickness, it describes or it defines how what is the spatial resolution of the imaging. So here is an example how different resolutions look like in fMRI. So on the left there is 3mm resolution. That is actually quite common in fMRI studies. But when you compare it to 1.5mm resolution, it seems that it's quite blurry, and with the smaller resolution we can get better specificity of the signal. And as I mentioned, nowadays we can even go to less than 1mm. So that will be at that level that we can even separate some areas within gray matter. So basically in fMRI we will get individual time series from each voxel in the brain. Here on the right you can see an example how the fMRI image would look like during the scan. The time between two neighbor time points is called repetition time, and it basically defines your temporal resolution. Next we are going to look at some signal and noise characteristics of fMRI. So what we have learned this far is that with fMRI we can get high spatial resolution and medium temporal resolution on the order of seconds, and also we can get whole brain coverage. That is beneficial, for example, if you want to study some subcortical structures or nuclei. For example, in MEG or EEG, we can usually measure only the cortical activity. So that's a benefit of fMRI. And as a downside of fMRI, it's basically indirect measure of neural activity, unlike, for example, EEG or MEG. But it correlates. The bold signal still correlates with the neural activity. Here are some sources of noise in fMRI signal. First, there is thermal noise. That is because of thermal agitation of charge carriers in subjects and MR systems. This type of noise, it doesn't focus on certain frequency bands, but it's more like a broadband signal. Then there is system noise and artifacts that can be caused, for example, because of scanner drifts. The scanner might be a bit unstable, or there might be some inhomogeneity in the field. And also when we acquire and reconstruct the images to be analyzed, there might be some artifacts related to that. Then there are two kind of noise that is caused by the subject: motion and physiological noise. That can be, for example, respiration or cardiac activity. Before we start the analysis of fMRI, we usually go through steps of pre-processing. Pre-processing assures that the assumptions of the analysis are met, and it also we want to improve the signal to noise ratio as good as possible. Here are some common steps of pre-processing that we will shortly go through. Not all of these are used in every study, and in some of the studies they are done in a different order, but it really depends on what kind of study and analysis you want to do. So most of the analysis, they assume that a time course represents always a value from a single location. So the same time course, it comes from the same voxel. However, even the best subjects, they usually move during the scan, and this happens especially with long scans, or if we have a special population to be studied, for example kids, or if the patients, if they have some condition that they cannot really control their movement, then the motion can be quite prominent. There are basically two types of issues related to motion. So the first issue is the voxel displacement. So when the head moves, the voxel that was there before, it's not there anymore. So this kind of displacement, this can be corrected by rigid body registration to a reference volume. For example, the first slice of the scan or average slice. So you will transform all the volumes to be in the same space. However, that's not really enough, because there can be some intensity changes. So let's imagine that you just gave an RF pulse to the brain, and then there is sudden movement. So now when you are reading the signal from the nuclei, they might be in different phase, or they might be coming from different voxel. So that affects the intensity of the voxel. In this kind of situation, you can use the motion parameters that you can get from the scan. So you will get 3 translation parameters and 3 rotation parameters across 3 different axes, and you can use those time courses, for example, in your GLM analysis as a nuisance regressors. So you can see how much those time courses affect your, or how much they explain the variance in the data. It is, however, important to note that the motion correction is never perfect. So the most effective approach is to minimize head movements during the scan. Usually proper padding is enough to make sure that the subject is comfortable and cannot move his or her head very much. Some people also use some customized head cases to prevent the movement. Then another subject-related noise source is physiological noise. So it's related to the cardiac and respiratory cycles of the body, and those can also cause periodic involuntary movement. Usually the frequency range of respiratory noise is or activity is between 0.2 and 0.4, and cardiac activity is typically a bit more than 1 hertz. It's worth to note that if you use very long TRs, there might be aliasing, so you would need to take that into account. This kind of physiological noise is usually localized near large vessels. And here is actually a study where they investigated where in the brain you can see the highest variance explained by physio factors, meaning respiratory and cardiac cycles. So it seems to be quite all over the cortex. Here you can see signal measured from the respiration belt. That is the red curve here. But in addition to that periodic respiration at around 0.2 hertz, you can see that the amplitude of this respiration also fluctuates. So we can also see some slow variations in this kind of systemic physiology, and that can be also happening in heart rate variability, and that can also have some specific frequencies. So it's good to take these kind of things into account when you measure the data. And importantly, these kind of signals might have similar kind of properties than as the signals we are interested in brain. So be extra cautious with this. Here is an example actually about how the respiratory variations can be very clearly seen in the voxel-wise fMRI time series. So how can we deal with this kind of physiological noise? There are several approaches. For example, you can use multivariate data-driven approaches, for example, independent component analysis, where you can extract the time courses and spatial maps related to those different components. And you can go through the components and see if they match or if they correlate with the physio signal. And then you can remove those artifactual components or components that you are not interested in. As with movement data, you can also use physio data in GLM analysis as nuisance regressors, or you can use some more advanced model-based approaches. Then some more pre-processing steps. This video shows how the slices are acquired. So in ascending approach, you start from the bottom of the brain and then you proceed to the top of the brain. And for example, here the TR is 2 seconds. So it means that the slice in the bottom of the brain is taken 2 seconds before the slice on top of the brain, and that's a problem if you want to compare different brain areas. So slice timing correction basically corrects this mismatch. It takes that timing into account. On the right, you can see a interleaved approach. So in that approach, you basically go through the space twice. You take first every other slice, and then you come back and they take another set of every other slice, and this makes this problem a bit smaller. Then there is also multi-band approaches, so you can, for example, take simultaneously slice from several locations of the brain that are separate, and then that shortens your TR. Then usually the images are smooth spatially, so as smoothing reduces some high frequency noise, it improves SNR and inter-subject registration. So what happens in smoothing is that basically you just replace each voxel value with the weighted average of nearby voxels. And this weighting is usually Gaussian, so the voxels that are closer to the target voxel, they get bigger weight than the ones that are farther away. Here you can see some examples of how or the degrees of smoothing. So you can see that on the right the brain looks very blurry, so too much smoothing is not good either, but you should find some compromise. Then if you do a group analysis, you want to compare brain activity between different subjects. And different subjects, they have different sizes and shapes of the head. So what we can do here is we can normalize all of them into the same template, that is usually the average brain or a template brain. So then you can, for example, average the activity between different subjects or compare the individual variation in different subjects. Then there is distortion correction. So in the external magnetic field, there can be inhomogeneities, and they can cause some distortions. And those can be dealt with taking a field map, which basically is an image of those inhomogeneities, and then we can use that information to correct the distorted image. Here is an example with a phantom. So on the left there is the original true image. Then there is field map of the external field inhomogeneities, and without correction we would get distorted image like in figure C. But when we take those inhomogeneities into account, we can unwarp the image, and that looks quite nice compared to the true image. Those effects are not usually that dramatic in the brain, but also it can cause some. It can make the human brain pictures also a bit better, as you can see here on the right. There is, in the anterior part, there is some distortions, and after unwarping it looks much better. Especially if you are interested in that area, I would do this. Then in the last part, I will show you some typical fMRI experiments and how to analyze those. So typical fMRI analysis, it relies on looking at the different contrasts between different conditions. For example, here the subject is being shown pictures at certain intervals, and then there is rest between them. So you can see that in one voxel the time course, it follows the task design, and then we would make some statistical test to see which voxels are actually changing statistically significantly in this task. A common way to analyze this kind of design is to use a general linear model. Basically, here is the equation for it, and y is the bold response from one voxel, x is design matrix, it contains the predictors or regressors that explain the data, and beta is the value that tells how much each predictor explains the data, and then there is of course a noise term. To form this x design matrix, we can take the task paradigm, so the alternating picture is on, picture is off, and we can convolve that paradigm with the hemodynamic response function to get the model response, and that basically simulates how the brain would or the single voxel would respond to that kind of stimulus. In GLM we take this model response and we can put them here as regressors of interest. We can have several predictors. For example, if we have several categories of a stimulus, let's say houses or faces or dogs, each of those would have their own regressor. We can also include those nuisance regressors, as I told you before, relating the motion and physio data, so we can see if how much they affect the, or how much they explain the variance in the signal. And as an end result, you will usually have a spatial map where you see the map of individual voxels, and it can be, for example, these beta weights for different categories, or it can be some T statistics or F statistics, or some kind of measure that tells how significant these changes are. Another completely different approach to analyze fMRI data is to look at the connectivity between different brain regions. So we can take one voxel from this area, one voxel from this area, and we can look at the basic Pearson correlation between them to see if their activity changes similarly in time, and that can tell you which kind of functional networks there are in the brain. Here are some functional networks that many studies have been finding consistently. These are actually from resting state data, so in this type of analysis you don't even need to have necessarily any task, but you can look at what kind of brain areas are connected functionally during rest. So what these experiments and analysis methods I show, they are only basics. There are lots of more advanced techniques and methods, and the fMRI field is rapidly advancing, and those advances give us tools to answer new questions about the brain as well. So here, for example, fMRI with higher temporal resolution can show some more detailed information about the functional networks, or higher spatial resolution can give us possibility to look at different layers within cortex once we get less than 1 millimeter. And also you can combine fMRI with lots of other functional neuroimaging modalities like EEG, MEG, TMS, or PET, or some animal electrophysiology. So those will give us more complete picture of what happens in the brain, and also they might help us to understand the fMRI signals better. Here are some common toolboxes that you can use for pre-processing and analysis of fMRI data. We all know FreeSurfer, and then there is also AFNI, FSL, and SPM that are probably the most commonly used. And here are examples of two references about books if you want to learn more about fMRI. And that's all I have today. So thank you very much, and if you have any questions, I'm happy to answer.